the name of the Father, and of the Son, and of the Holy Spirit. Amen. Lord, grant us concentration that we may learn, listen, understand, have a peaceful mind, and remember that you're with us always. Amen. For this video, we're going to consider the shoot structures and start with an overview of the external shoot structures, and then begin to look at some of the modifications that were involved with that. So here we see both two examples of shoots, one that has the leaves on it, one that's in sort of the sessile or the not actively growing stage of the plant growth. And so when we talk about this, we're looking at, this would be the external structures. You can see the nodes that are regularly placed along there, and they form these iterative units, as I've already mentioned, that go along there. With each of the nodes, we can see associated with that two things. One is the growth that's occurring out of that. In this case, we're talking about a leaf that's growing out of the stem. But we also have an auxiliary bud that's present with that. And the auxiliary bud is what's going to provide for the growth after the leaf has been removed. So in the fall, when trees lose their leaves, they'll use the auxiliary bud in the spring to begin to grow a new leaf or a new branch out of the stem itself. In addition to that, we can also notice here at the end of the stem is what we refer to as the terminal bud. And the terminal bud you can think of as being similar to the location of the plant apex. This is where the primary growth is going to occur. And so the primary growth will occur out of the end of that. Now, it's important to note that even a plant that has gone into a dormant stage, for example, the trees after they lose their leaf, at the location of the previous leaf, we have what we refer to as the leaf scar. And this is not an insignificant structure. You'll notice at the end of the leaf scar, we have the bundle scar. These are where the vascular tissues that were providing nutrients and water to the leaves were located. In addition, the leaf scar is an important source of hormones. Okay, so it's going to actively be producing signals that's either going to prevent the growth or, as it begins to stimulate in the spring, to begin the growth of a new year. Okay. If we look at the internal structure, again, we've already kind of looked at this, but the internal structure of the stem, the shoot that's growing up, we can see again a difference between the eudicots, again, a eudicot or true dicot. These are going to be the plants that are growing with two embryonic leaves, and will show the ring-like structure of that, which will allow for the secondary growth to occur, the rings on a tree, for example. Inside of that, we have internal ground tissue, referred to as the pith. This is the woody-like material. Likely a lot of the sclerenchyma cells would be there, the cells that would contain lignin and other strong support material. Outside of that, we have the actively growing ground tissue, or the external ground tissue, which forms the cortex around the outside, with the dermal tissue on the outside of that. With the monocots, or those plants that have one embryonic leaf, think of grass. Grass is a good example of a monocot, and we'll come back to that example. But we don't see the same type of structure where we see sort of a random or a scattering of the vascular bundles. and the ground tissue is mixed with that. So it's not the same type of organization. So when we talk about modifications of the shoot structures, keep in mind that this type of structure will have an important consideration there. So what are some of the considerations? Well, secondary growth is an important consideration. And this is only shown in the dicots or eudicots as a result of them having this ring-like formation of the vascular tissue. And so each year that it grows, a tree will put out another set of vascular tissue that will grow and form the rings. Monocots do not have this regular formation and therefore lack the ability to show secondary growth. As far as modifications, we have 6 different modifications that will be present here that we can talk about them. The first one are bulbs. Bulbs are not a root modification. Bulbs are a leaf modification that grows below the ground. And so, to use one of my favorite analogies, as Shrek would say, onions have layers. And the reason they have layers like that is because these are leaves or stems that are growing very close together underground and form the bulb. Growing out of that bulb are adventitious roots. Again, these are roots that are growing directly out of the shoot. So these will be connected to the stems. A second modification are the rhizomes. And rhizomes are going to be the stems that grow underground horizontally, and they're going to have scale-like leaves that will come out at regular intervals. Okay? We will see that underground as these scale-like leaves, which will eventually grow up through to form this photosynthetic leaves that will grow out of the ground. So rhizomes are going to be stems that grow under the ground near the surface of the ground that will allow for the continued growth. And we see these if you know what a tiger lily is. A tiger lily is a good example of this that will grow under the ground. Similar to this, and I'm going to give you a term because we're going to use this a couple of different ways, okay? And that is a stolon. S-T-O-L-O-N, okay? And stolons, okay, are sort of these long extensions that will grow out of the stem itself, out of the shoot itself. And some of these, for example, can form runners. And so if you've ever seen a strawberry plant, you know strawberry plants will put out these long runners which can take root at different points to be able to extend and grow. In this case, the stolon is going to form a new node further down, which is going to form another plant that can grow. Another modification of a stolon is a tuber. Okay, and so tubers, potatoes, are actually stem modifications. They are a stem modification for the storage of carbohydrates. And so the stolon will grow out, it grows underground, and then the end of the stolon, or the tip of the stolon, will become swelled where the carbohydrate storage will occur. And so potatoes are an example. And so what do we see on potatoes? Well, there's no growth rings. So we know that it's not a root modification, but potatoes have eyes. And these eyes are nodes. And so the eyes give you an evidence that this is a stem modification and not a root modification. So tubers are a good example of a stem modification. These are the stolons that have been modified to be able to store carbohydrates. Other plants will modify their roots to form tendrils. Again, very similar to runners, but rather than forming nodes that will then form new plants, tendrils will be used to be able to wrap around structures to provide support. A good example of this is found in grapes. If you've ever seen grapes growing, you know grapes have these arbors or these wires that they'll grow up, and as they grow up they put out tendrils which will attach them to that. And so tendrils are going to allow for the connection or support of the plant. The last modification in this group is a cladophyll. Okay? And cladophylls, they're found in cacti. Okay? These are really a flattened stem structure that make up the structure of the cactus. They may look like leaves, but they're not actually leaves. If you look on the surface, it's the spikes on the cactus that are actually the leaf modifications. So cladophylls are a photosynthetic modification or flattening of the stems that will allow for conservation of water, to prevent water loss, and they're also going to be modified in a lot of cases to store water. If we're talking about the shoots, we can't forget to speak of maybe the most obvious modification or the most complete, and that is the leaves. Okay, so leaves, we could call them, as I have on this slide, the photosynthetic organ of the plant. So the leaves are going to be collecting, they'll have a large flat area, which we refer to as the blade, that's going to collect sunlight and use that energy for photosynthesis. It's connected to the plant through a structure called a petiole. And the petiole is just basically the stem of the leaf, which will connect to the stem of the plant. And that's where the vascular tissues are brought into the leaf to allow delivery of that. When we talk about leaves, we first need to classify the type. Okay? And so there are two different types. There are microphyll, which are not very common. Okay? And these are leaves which will only contain one vein, and that vein does not extend to the end of the leaf. So they're very small leaves. They don't have a complete structure, and they're going to only be very small. So they're not a very common type. The more common type are referred to as megaphyll. And so most plant leaves you see will be megaphyll. And these are leaves that will contain multiple veins, and those veins will extend all the way out to the end. Well, what are the veins? So the veins are the second part. The blade and the vein make up the second part. Veins are the vascular bundles of the leaf. So this is going to contain the phloem and xylem that will be delivering water and minerals and also carrying away some of the sugar that's being produced. And we can see again, another distinction that we can make about the leaves is the type. Monocot leaves will have parallel vines. So on the bottom here, you have an example of a monocot. They have parallel veins that will run through there, and for the most part, these parallel veins will lack a true petiole. They're connected sort of through the whole plant. Again, I used this example before. Think of grass. The leaves of grass will contain parallel veins that are coming out, not a true stem, so there's no true connection in there. Eudicot leaves, on the other hand, true dicot leaves are going to show net-like veins. Okay, and so you look at the close-up of the leaf here, you can see the network of leaves that come out of there and the branching of the vascular tissue which is going to increase the efficiency. So it's going to be more efficient in its process of delivering the nutrients and collecting the sugars. So we can see with leaves, just as we can talk about the stems, there are different types of leaves and some modifications that we can see. So the first thing we should talk about is a simple versus a compound leaf. A simple leaf, the blade is completely connected and the veins run out through there. So these are going to contain many lobes. Think of oak leaves I have on here. Think of maple leaves. They have one vein or one blade that connects all the veins together. Compound leaves, on the other hand, such as showing here, are made up of multiple little leaflets that are connected together by a common vein system. And so we see compound leaves, smaller leaflets, not a vein that makes up the whole thing, that's going to be on walnut trees, ash trees, box elders, for examples there. We can also see a modification of the leaf structure based on the thickness or the properties that are present there. So as a quick overview, we can refer to leaves of having a unique stratification. That is, if we look at the different layers from the top to the bottom, we can see that they're not the same on the top and the bottom. We'll notice that on the top, there's a stronger or thicker cuticle layer that's going to be present there. And this is the waxy covering that will prevent loss of water. Below that, we have the dermis layer, and you'll notice there's an upper dermis and a lower dermis. These are the dermal cells, and the dermal cells are pretty much clear. They don't contain a lot of chloroplasts. They're not the photosynthetic cells. That's going to be found underneath. And the photosynthetic cells are below that, so these are just protective cells. On the bottom layer, we have one additional type of dermal cells, and that's the guard cells. The guard cells that are on either side of the stoma. Stoma are only on the bottom of the leaf. Okay, so below the upper epidermis, we have a layer of cells, ground cells, that are very tightly packed together. These are referred to as palisade cells. They're pretty much all made up of the chlorenchyma cells. The chlorenchyma cells are parenchymal cells that contain chloroplasts. And so they're tightly packed together, they're collecting the sunlight. These are the photosynthetic cells. Below that, we have a more open area. Okay, we can see in both of these pictures that area there. And that's made up of the spongy mesophyll cells. And the spongy mesophyll cells are going to be less tightly packed, a lot more air spaces open in there. And so this is going to be where we see gas exchange occurring. So gas exchange occurs in there, and of course the bottom lower epidermal layer. So some leaf modifications then. So for the leaf modifications, we can talk about floral leaves, the poinsettias you have out at Christmas time. The red leaves on there are leaves. They're just modified to attract insects for pollination. The spines or the spikes on a cactus, these are modified leaf structures that have reduced surface area to prevent water loss. Another different we can talk about are shade leaves. So if we look at this picture here, you can see these are leaves collected from the same tree. This one would be a top leaf exposed to a lot of the sun, which means that it has a smaller surface area. It has access to a lot of sunlight, so photosynthesis can occur regularly. The leaves lower on the tree, or the shade leaves, are going to have a much greater surface area to be able to collect more sunlight. We also see that if you look on this, the shade leaves are thinner. They're not as thick. So they're going to be less thickness, not as much concern about overheating, so they don't need as much mesophyll tissue to be able to protect it. The last leaf modification here that we can talk about are insectivore leaves. Okay, and the insectivore leaves, the pitcher plants or the Venus flytraps, these are leaves that have been modified in order to collect insects. Insects are a great source of protein, ammonia, and so therefore some plants will use this to be able to supplement their needs. Okay. The last thing that we want to talk about real quick with respect to shoot modification is going to be the C3 versus C4 plants. And if you remember C4 plants, there is a spatial separation between the process of carbon fixation as it relates in the C4 plants to Pep-C or phosphoenolpyruvate carboxylase, which is going to be present in the mesophyll cells on the outside, and the cells that are in the center, which are the bundle sheath cells, which is where the Kelvin cycle is occurring. And so these are plants that are running for a longer period of time with the stoma closed, and therefore they don't have as much of a concern about the buildup of oxygen and rubisco with its trouble. In the C3 leaves, you can see the mesophyll cells are all throughout. The bundle sheath cells are much smaller because we're not separating the process. So this shows you, again, how we can see a difference in the leaves based on the type of plants. Thank you for watching this video again. If you have questions or comments, please post them on the Schoology discussion board and let me know if you have any questions. Thank you.